Good morning, everyone, and welcome to the third and final day of the inaugural Intersection Scholars Symposium. Thank you all for your enthusiasm and support these past few days. We have heard some phenomenal talks and are excited for the fellow research talks in neuroscience session. I am Dr. Lillian Brady, an Academic Pathways Postdoctoral Research Fellow in the laboratory of Dr. Aaron Calipari at Vanderbilt University in the Department of Pharmacology and the Vanderbilt Center for Addiction Research. I will be co-moderating the fellow talks in today's neuroscience session with Dr. Yunlu Zhu. Dr. Zhu is a Leon Levy postdoctoral fellow in the laboratory of Dr. David Shopik at NYU Langone in the Neuroscience Institute. We have an exciting lineup of research talks today from some talented postdoctoral fellows who are sure to be future leaders in the neuroscience field. Each presentation will be 15 minutes long, and there will be a 5-minute question and answer session after each talk. We ask that you type all questions into the Q&A box for Dr. Zhu to ask the questions to our presenters. Please follow the link in the chat to give feedback to the fellows on their talks. The same link can be used for each fellow in this session. At the end of this session, we will show the QR code to access this, and at the end of the sessions as well. The first talk is by Dr. Josefina del Mármol. She earned her PhD in molecular neurobiology and biophysics from the Rockefeller University and is currently a postdoctoral fellow in neurophysiology and behavior at the Rockefeller University in the laboratory of Dr. Vanessa Ruda. She has an impressive record of mentoring and outreach and has received a number of fellowships and awards recognizing her research. Her talk today will focus on the structural basis of odorant recognition in insects. And so with that, I would like to welcome Dr. Josefina del Mármol.
All right. Thank you, Dr. Brady, for that wonderful introduction. Let's see. Can you all see my screen? Hopefully. Okay.
You can see it.
Okay. I want to thank you also, the organizers of this fantastic symposium that has been a great resource, and I hope it becomes a staple in the years to come. I've been interested for a long time in the molecular mechanisms of sensory detection, and today I'm going to tell you about my recent work in the lab of Vanessa Ruda, trying to understand the logic of chemical detection in the olfactory system. The olfactory system faces a unique challenge as a sensory modality because the chemical world is remarkably complex. The chemical space is composed of hundreds of thousands of different molecules, and often what we think as a distinctive smell, like the smell of coffee, is usually comprised of hundreds of different compounds, and I'm only showing a fraction of them here. And so the extraordinary task of the olfactory system is to detect these compounds in a way that allows animals or the olfactory circuits to make sense of this information. And this challenge is unique among sensory modalities. For example, vision starts with a light wave, and so we can use quantitative metrics to describe visual stimuli like wavelength, and this directly captures a percept that we perceive as a hue. And so in humans, every color in the visual spectrum is encoded by just 3 receptors that are able to detect the wavelengths of light that seem sufficient for our ecological needs. Olfactory stimuli, however, varies discretely along many different axes with no single dimension that can describe the world of odors. And so, and so how do olfactory receptors cope with this diversity? And one possibility would be to employ a single receptor for each type of molecule, but to detect hundreds or thousands of different molecules, this is likely unattainable. And instead, animals make use of a comparatively smaller number of receptors, most of which can detect a variety of different molecules, and they overlap in their receptive fields. And so this hugely expands the chemical space that we can detect, but has an immense implication at the molecular level, because it implies that receptors detect a wide variety of different molecules. And how this has been achieved at the molecular level, we don't know. We fundamentally don't know if this is achieved through a single binding site that is very promiscuous or through many different binding sites. And if there is a shared binding site, what is the molecular logic that determines the type of chemical compounds that a receptor can detect? Now, however complex this strategy has been successful, as animals across the tree of life have converged on this strategy, even using different types of molecules or families of receptors, like insects that use ion channels and mammals that use GPCRs. And so while animals seem to be able to make do with only a handful of photoreceptors to detect light, they have evolved large and highly diversified families of olfactory receptors that are uniquely tuned to detect the chemical cues of their ecological niches. And so much like our vision is determined by the type of photoreceptors that we have in our eyes, the olfactory capabilities of an animal are determined by the type of olfactory receptors and their interactions with the ligands. But unlike for vision, we don't have a structural understanding of the mechanism of odor recognition, as of yet there is no atomic resolution structure of an olfactory receptor, an odor activated olfactory receptor from any species. And so in the Rural Lab, I have been working on obtaining a structural understanding of the molecular mechanisms of odor detection. And I focused on insect olfactory receptors because the sense of smell plays a key role in the ecology of insects. For example, mosquito vectors of disease and several agricultural pests, they rely on their olfactory receptors to detect their targets. And this has devastating consequences for human health, for public health and the economy. And if we understand more about the way olfactory cues are recognized, we can develop new strategies to control pests and insect vectors of disease whose diseases kill almost 1 million people yearly. And one of the reasons we don't have structures of olfactory receptors yet is because they are notoriously difficult to manipulate in vitro. And in the case of insect olfactory receptors, this is in part due to the fact that they are heteromeric, with a olfactory receptor subunit, a proper olfactory receptor subunit that binds odors, and a co-receptor that is inert, does not bind odors. Now, this had added a layer of biochemical complexity. But recently, the genomes of more basal insect species like the jumping bristletail were sequenced, suggesting that there are species that have only olfactory receptors and no co-receptors. And so we reasoned that if these are indeed odor-gated ion channels or receptors, they can offer an inroad into understanding the molecular mechanisms of odor detection. And so I began by deorphanizing these receptors from the jumping bristletail, measuring the response to a panel of 54 chemical molecules using an in vitro fluorescence calcium assay. And so on the top, you can see the tuning curves for 2 of these receptors, OR1 and OR5. And on the bottom are the dose response curves for some of their ligands. And you can see that these receptors indeed respond to odors with distinct ligand specificities or odor specificities and tuning breadths. But furthermore, I found that this receptor here, OR5, it is activated by many structurally diverse compounds that share no obvious structural or chemical feature. And this includes canonical odorants like eugenol or limonene, but it also includes DEET. Now, DEET is the potent insect repellent that, while it has been long speculated to bind olfactory receptors, it has never been explicitly demonstrated. And so I focus on OR5. I first used electrophysiology to verify that it is indeed an odor-gated ion channel like all canonical insect olfactory receptors, which it is. And so I chose OR5 because it allows us to ask, how can a single receptor accommodate this chemical diversity? And so because of the biochemical tractability of these receptors, I was able to obtain the atomic resolution structure of OR5 using cryo-electron microscopy. Now here's the cryo-EM density, and first you see the outline of the detergent micelle, and inside sits the channel that assembles as a tetramer. The majority of the protein is embedded in the membrane. All 4 subunits are assembled around the channel pore that we presume opens and closes in response to odor binding. Now this structure offered a lot of interested insight into this family of channels. And for example, it confirmed years of experimental evidence from our lab and other labs that suggest that despite the large sequence variation, this family of receptors retains a high degree of structural conservation. But today, what I'm more excited to talk to you about is the chemical basis of odor detection. So to do so, I first saw the structure of this receptor unbound and in complex with the other eugenol. So here's the unbound structure in a side and a front view. And here in the side view, I remove the front and back subunits so you can focus on the pore. In the unbound structure, the pore is constricted at the top to a diameter of 5.3 angstroms. And here is the eugenol bound structure. And we can clearly see that the pore dilates to a diameter of 9.1 angstroms. And now we know that this is a cation channel, and so these diameters will be consistent with this transition corresponding to the closed and open states. And so now we can go look for the other binding site. And so here I'm looking at the bound structure from the top, the eugenol bound structure. And if we take a slab near the extracellular leaflet in the transmembrane domain of each subunit, we find this cavity. And here's where we find the density for eugenol, which is of the expected size and shape, and it allows us to place the ligand in that site. And so let's zoom in into the pocket to take a closer look. And I'm here zooming in into only one of the subunits. And here I'm showing you the residues that are in close contact with eugenol. And what is immediately apparent is that the binding site is this beautiful hydrophobic box that is organized around the density for eugenol. And most of the interactions between the ligand and the residues are hydrophobic. And in data that I don't have time to show you today, I've mutated all these residues and I functionally validated the location of this binding site. And so we've located the eugenol binding site, but now what we want to know is, can this pocket accommodate the other ligands that activate the channel? And so for this, I first turned to molecular modeling, and I first found that eugenol is predicted to dock in this exact same position very favorably, exactly what I found in density, which is a great validation. But then I also found that other structurally diverse compounds that I showed you before, like octanol or limonene, geosmin, ethyl butyrate, and essentially all of the ligands that activate this receptor, they too can dock in this position, all predicted to interact through hydrophobic interactions with the surrounding residues, just like we observed experimentally for eugenol. And so this would suggest that all these ligands share a binding site. But we want to make sense of this chemically. And I told you earlier today that the activating ligands don't share any immediate obvious feature, like the chemical group or the molecular size or the shape or and so we wanted to find something that they do share, and they do share a low polar surface area, a high partition coefficient, and a low hydrogen bond count. And these features are a great match for the pocket that we found, because the pocket that we found is essentially a hydrophobic cavity that can stabilize hydrogen bonds and or other polar interactions. But then I can do something else to add evidence to the shared binding site, and I can solve the structure of this receptor in complex with something else. And so that's what I did. I'll solve the structure of this receptor in complex with DEET. And so first I found that the DEET bound structure is also open, which you can see here in the left. And so where is DEET? And well, the density for DEET is in the exact same pocket where we found the density for eugenol. And the DEET density is of the expected size and shape for DEET. So we can now, with a fair amount of confidence, say that this is a shared binding site, at least for eugenol and DEET. So now that we can visualize this receptor in presence of two different ligands, we can also start thinking about how we can accommodate these structurally diverse compounds. And we see that the structure of this pocket is very similar in the presence of eugenol and DEET, meaning that there is no major rearrangement that seems necessary to fit these ligands, but there are slight differences that allow us to make functional predictions. Specifically, in the presence of DEET, we see that this methionine here, it needs to move out of the way to allow for the density of DEET that is larger and protrudes further into this region. And so if we mutate this methionine into a smaller residue like valine, as we will predict, we see an enhancement of the affinity and efficacy of the larger odorant DEET, and it decreases the affinity of eugenol. And so one of the interesting implications that broadly shapes the way we think about odor detection is the fact that my data suggests that all of these ligands bind in the same place, which means that any small mutation or variation in the binding site should affect the activity of most of the activating ligands at least. And that is indeed what we see. We see that this small mutation differentially affects the affinity or efficacy of all the panel of odors. And so to summarize, I want to highlight that this first structure of an OR provides us with initial insight into the determinants of olfactory tuning, but naturally this one structure is not the entire story, as there are olfactory receptors that are incredibly selective for specific cues like pheromone receptors, and there is olfactory receptors that are broadly tuned, but to a very different set of compounds. And so we want to know how that happens, and I can use structural biology now to obtain a broader understanding of the logic of chemical detection. Now, we think that this pocket we found is relevant to other receptors because going back to the past 20 years of research, most of the mutations in polymorphisms that have been described to alter odor specificity, including those that affect the sensitivity to DEET, they map to this pocket. And this offers a window into understanding the structural basis of behavioral evolution, because insects have evolved olfactory preferences by rapidly diversifying their olfactory receptors to detect new or newly relevant cues. And that ties the evolution of the receptors with the emergence of species-specific olfactory adaptations. And an immediate example of that and an immediate target would be mosquito vectors of disease, because they have recently evolved a preference for human volatiles largely by tuning their olfactory receptors. And so I want to gain a structural understanding into the ways by which mosquitoes are able to hone in on human volatiles, which has extensive implications for drug discovery. And lastly, we also see how the response to dozens of odorants are highly intertwined by the shared binding site. And this is an interesting challenge in the context of the combinatorial encoding of odors. And in the long run, it would be really interesting to study how small variation in receptors can alter the representation of odors in the animal. And with that, I would like to thank you all for your attention, and again, the organizers of this great symposium. And I want to thank a number of people, the funding sources, especially my mentor, Dr. Vanessa Ruta, for outstanding personal and scientific mentorship, and a brilliant research assistant, Kenzie Yetlin, soon to be grad student, members of the Rural Lab, and Leslie Vostal for advice throughout, and the members of the cryo-EM Research Center that helped enormously collecting data during the pandemic. And happy to take questions. And I wish I could see you all again.
Fantastic talk, Josefina. And the floor is now open for questions, and I can see questions are pouring in. So Sol Lacos asked, from your work, can you predict what other components can activate the receptor?
Yeah, no, I'm not showing my screen anymore, right?
No, you're not.
Sorry. Can you predict what other compounds? Yes. Yeah. So to an extent, yes. And that is actually a beautiful part of having this structure, which is that you can do in silico screening, and it will give you leads. And with in silico screening, you can find millions of compounds that you have to sort of you have to bring into the bench later on. But one of the things about olfactory receptors is that we only know the receptive field through experimental determination, like what I did. You expose them to whatever 50, 100, or as many as you can, but you know, we can't sample experimentally millions. And so these structures will expand our ability to sample, at least in silico, a large library that we can then bring into the bench. And actually, one of the compounds that I showed you before, geosmin, it came out of a little sort of trials screening that I did.
We have a question from our fellow, Lauren. Can you predict competitive inhibitors or mutations that lock the receptor into closed conformation?
Yeah, so there's two things into that. That is a little bit into the mechanism of how the binding site translates into the channel opening. And that's something very interesting to us. And we have some insight into that. And there's definitely mutations that we found that can significantly impair activity. Yeah, we're sort of working on that.
We have a question from Stephen Blacklow. Lovely seminar. How does the compound gain access to the pocket if it is inaccessible from solvent? And a related question, what are the kinetics of binding, and how do the kinetics of binding compare to the kinetics of channel opening?
Yeah, okay, so the the pocket is in the transmembrane region, but it's relatively close from the relatively close to the extracellular side. And we don't see a loop that we believe is sort of like looping inside. And there is a number of possibilities. So one thing is that, remember that in vivo, volatiles are likely bound to at least olfactory binding proteins or other ways in which they can partition into the extracellular solution. And so whether they are, how they're shuffled through to the receptors and whether they go through the outside or through the membrane is something that we're trying to investigate. And we can do this with electrophysiology. We can do patches and see if it goes through the membrane or if it goes through the inside or the outside, and we're at it.
We have a few people asking that, do you have any insights into how those molecules get into the pocket?
Yeah, so we're getting at, but definitely it's not so far from the outside, and there is a mobile part sort of sitting on top. So there is definitely the possibility that it comes from the extracellular side, but it could also be partitioning into the membrane, and we're getting at that. Yeah.
All right, so we still have a few questions unanswered, but for the sake of time, we have to move on, and all unanswered questions will get copied and sent to the speakers. Thank you.